bit about what are stable isotopes. And we can imagine that we can go out in the field and collect phytoplankton or zooplankton or fish or bird tissues and measure carbon and nitrogen in pretty much almost any of the tissues. So this could be a feather or blood or muscle. And if we look at the ratio of the heavier to the light isotope, we can know something about whether an individual was feeding at a higher trophic level or at a lower trophic level. And then for carbon, the variation in carbon can tell us something about the source of primary productivity. So, for example, was the phytoplankton consistent with like an offshore plume or a more inshore plume? And so that's sort of the basics of how we use isotopes, and it's a really cool technique because we can look at relationships of how the isotope signatures are associated with the ecology of the animal. And so if any of you probably don't know how exactly we do it in the lab, but we'll take basically little ground up fish and stick them in these tin capsules. So these are really, really small samples that we put in these capsules and then keep them organized in these 96 well plates, and then we send them off to the lab up in Fairbanks at the University of Alaska, and they combust them in their mass spectrometer, and then they send the data back to us. So it's kind of how we do it. And so we looked at this in the herring that we've collected from the sound, and basically what we found is that the delta 13C variability really does correlate with energy content of the age 0 herring. So it's kind of a lot to digest, but these are our fish from November and our fish from March, and you can imagine we have our carbon gradient here, which is Gulf of Alaska sources on the left and Prince William on the right. And so for the November fish, we see this really strong relationship where if you're depleted in carbon, you tend to be more energy dense as a juvenile herring in November. And that's also true in March, but the relationship isn't quite as strong in March, and that's probably because a lot of the fish haven't been foraging over the winter time. So what's really cool about all of this is that we're starting to learn that these carbon-energy relationships really occur across a spatial gradient in Prince William Sound. So what I mean by that is we have these sampling bays, so we've got these 8 bays, and then sometimes we sample in Cordova Harbor. And basically what we see is the carbon-energy relationship kind of organizes itself by the west side of the sound, and then the middle part of the sound, and then the eastern part of the sound. So just keep in mind these different colors as we look at this next graph. So basically, these are the data here, where we again have carbon variation here on the X with Gulf of Alaska carbon on the left and Prince William Sound carbon on the right. Energy density on the y-axis, and you can see those west side bays sort of segregate out here as high carbon and high energy density and more depleted carbon, whereas the east side tends to be lower energy density and more Prince William Sound carbon. So what's neat about this is that we think this variation in carbon and energy is really being driven by the circulation in Prince William Sound. So this is kind of a complicated graph of Prince William Sound, but this is basically the sound here, and the boxes are around the entrances of Hinchinbrook and Montague. Basically, we think the majority of the Gulf of Alaska water is coming in through Hinchinbrook entrance and then moving around the sound this way, where it's very deep trenches along the west side. So you can imagine that it would make sense that we would see more of a Gulf of Alaska signature on the west side and not so much on the east side. So we think that these Gulf of Alaska processes are really important in juvenile herring growth and survival. And there's some other data that contribute to that conclusion, which is some of Scott's work, where he's looked at age 0 herring growth and the diatom anomaly. So here on the x-axis is diatom abundance anomaly, so we've got more diatoms and less diatoms. And then this, the y-axis is first year growth increments. So this is based on the scale growth data, and I'll talk a little bit about that. And so you can see that fish that grow more are years that had fish that grew more were years of higher diatom abundance in the Gulf of Alaska. And so I actually just find this super fascinating. So we could take scales of herring and actually look at the growth increments over specific years, sort of like a tree ring. And what we're basically measuring in this graph is the first year growth, which is basically shown here. So you can sort of see these rings that have grown on the scale. So this is actually a 6-plus-year-old fish. Obviously, the fish that we catch as juveniles wouldn't have this many rings, so we can measure this first year of growth in the fish that we catch. Some other work by Fletcher down with Noah has shown some really strong correlations between young of the year pollock and herring recruitment. So the herring is in the black solid line and pollock is in the more dotted line. And so you can see over time there have been these spikes of big recruitment that seem to be consistent between pollock and herring, which just sort of lends itself to this idea that both species are responding to some sort of large regional driver that's contributing to greater recruitment. So back to our sort of conceptual model here. We've been talking about sort of the provisioning stage, and some of the conclusions were that growth of the fish is emphasized until about 80 millimeters, and then after that, the fish are increasing energy energy allocation to lipid stores. After they switch, the faster growing fish get fatter, and that lipid and growth are influenced by Gulf of Alaska processes, and then it seems like the western districts of Prince William Sound are probably pretty good hotspots of herring production, at least regionally. So I'm going to go on for the rest of the talk and talk about some of the fasting side of the story. And here, what we're interested in is what happens during the winter when the fish are fasting. And so what we ended up doing was sampling nursery bays in March, so this is after the winter time. And we also conducted a high-resolution study between September 2011 and June 2012 in Simpson Bay, where we actually did monthly sampling and assessed growth, stored energy, and size. Yes, we just did the high-res stuff in Simpson Bay for 1 year, whereas normally we just have this time series of November and March. And so basically, this is what happens in the wintertime. We go out, measure them, catch fish in November and March, and look at their body composition. And so the November data are in gray, and then the following March is in blue. So you can just kind of forget about this first year. This would be the first November in the time series, and you can see every November fish are much more energy dense, and then we get to the spring and they're much more reduced. And since about 2007, we haven't seen a tremendous amount of variation in these data. I mean, there is a little bit. So fall 2012, the fish were in very good condition, and then following that spring, they were in much better condition than some of the other years. So that's what some of the basic data look like. And then what we were able to do with the high-res study is really look at variation in energy density at different monthly intervals in Simpson Bay. And so this was kind of cool because we were sort of always assuming that the loss of energy over the wintertime was linear, like it was just consistent each month. But actually what we found was that fish are building energy stores up till November, and then they drop down really quickly by December and just maintain this very low level through the wintertime until they're able to start feeding well again by April. So this was actually a little bit of a different model than how we thought the system was working. But what's also interesting about this, and this is again some of Fletcher and Ron's data, is that they found that small fish maintain energy, but large fish lose energy. And so what we're looking at here is the length of the fish, so the size of the fish versus the total energy of the fish for fall in blue and spring in red. And so what you can see, and I think we sort of see it better in these years, is that the small fish, their total energy is pretty similar. But when you get to these larger size fish, that's where you see more variation between energy status between fish. So the large fish tend to lose more energy over the wintertime than the small fish. And so, the other side of the story is that small fish also become rare over the wintertime. So again, these are the data from Simpson Bay over the high-res study. And what we're looking at is variation in fork length over the wintertime. And what we see is that by, March and June, the small fish have dropped out of the system. And so basically, this is just another graph that shows that same dynamic where we're looking at fork length and just the number of fish for November in red and March in blue. And so, a lot of the small fish are here in red, and then the purple color is sort of the overlap between November and March, and then these larger fish are around in March. So the question is, what's causing this we think it's size-selective mortality over the wintertime. And part of the story is that when we look at the lipid content of the fish that have a very low lipid level have a disproportionately high types of prey in their stomach. So the stomach fullness is increased in fish with low lipid levels. So this suggests that the fish that are not in very good condition are actually going out and foraging in the wintertime, whereas the fish that are in better condition in the wintertime, they don't forage. So we think that part of what's driving size-selective mortality might be predation on these fish that are exposing themselves because they need to go out and forage. So, some other data that lend to this idea that fish that are foraging are actually eating. These are also some of Fletcher's data, and we're looking at the fall type fish and the spring type fish. So the fall fish that are more energy dense in comparison with the spring type fish actually have a different fatty acid signature, and so that's what we see between the blue and the red here. So the fish in the spring are foraging, and that's what's driving their change in the fatty acid signatures. So then this kind of brings us to the predation side of the story, and Mary Ann's done a lot of work on bird predation of juvenile herring, and actually what she calculated was that marine birds in the Sound are consuming 1,500 tons of juvenile herring per winter, or about 213 million fish, roughly, between January and March, which is a lot. We've also seen that groundfish might be playing a role as an important predator of herring. So herring in pollock stomachs, and this was in November 2010 and 2011, tend to be on the smaller size than the larger fish. And there was a significant amount of herring in cod stomachs as well. So we think groundfish and seabirds are certainly really important predators of fish that might be taking fish that are smaller that need to go out and forage more in the wintertime. So kind of the conclusion, sort of wrap up here in the ecological factors that we think is influencing juvenile herring survival. One thing is that oceanographic conditions in the fall are probably playing a real important role in regulating the provisioning of the fish. There's strong connections to the Gulf of Alaska carbon that lead to better provisioning. Small fish with low lipid levels spend more time foraging. Predation risk is elevated for foraging individuals. And so basically, winter severity is really a function of herring size and prey availability and also predation risk. So just to kind of wrap up, I just wanted to give a lot of thanks to a number of people, mainly the trustees at EVOS for funding the work, a number of technicians that worked at the Science Center, so Megan Roberts and Julia McMahon and Vivian Gonzalez and Austin Potter were working with us last summer. The crew of the Montague, because they helped us collect fish in November, and then also fishermen and staff at CDFU for helping us with the spring collections. Steve Moffett with Fish and Game here in Cordova has let us use his scale to weigh out our isotope samples, and then also Norma and Tim at the UAF Stable Isotope Lab learning our samples. So think that's all I got.
Sounds like a starvation model.
So what about winter water temperature?
Good question, which I didn't say on there. So the question is, what do water temperatures have to do with herring? And we think that herring don't do very well in warm water temperatures. So actually, go back here. Hey, Meadow, can you put background up? This one with the lots of bar graphs is number 31. So this is the long-term time series, and we think that 2012, 2013 seemed to be a pretty good year for the condition of the fish, and it was associated with cooler water temperatures.
Didn't we have a late spring in 2013? 2012 or 2013?
This winter's been really bad for herring, and it's obviously been really, really warm. So, yeah, you might think that the fish have an easier time if it's warmer, but I think in this case, herring aren't responding that way.
If the waters are getting warmer, is there a chance they will move up to colder regions like the sun?
I think that's definitely a possibility. We're trying to understand better the population structure of herring, so whether at a genetic level, Prince William Sound herring are related to southeast herring or herring found further west. And that would allow us to try to understand whether fish are dispersing, to have the capacity to disperse to these other areas. I don't actually know if these sound herring are moving or not. I think
So the genetic result is that Yakutat, Kayak, and Prince William Sound, the genetics were all similar. Cook Inlet was totally different. And then as you go southeast, they definitely differ. But the real surprising one was that if you hit Cook Inlet, you're just talking a totally unrelated fish.
Any other questions?
So how do herring overwinter actually? Are they overwintering at certain depths or different areas?
Yeah, so let me see if I can. Sorry. Meadow, maybe you can grab one of the figures of Prince William Sound.
Is it earlier or later? Oh, a map, you mean?
At the beginning.
Here? Yeah, that would work. So we know that the juvenile fish are basically in these nursery bays. And one thing that's been really hard for us, because we do have a component of the program that uses acoustics to try to look at the biomass of the fish, but a lot of them are found really near shore, like too close to the shore for the boat to actually work in. So we think that they're using a variety of different habitats, I guess is my point, in a number of nursery bays. And they're seeking out areas maybe that they could hide under ice in the wintertime and avoid predators, things like that. But obviously earlier in the year, they are free-floating larvae that eventually settle out in certain areas. And we think that where they settle out has a big role in the type of condition they end up in through the wintertime, meaning that we think conditions on the west side of the sound with the carbon gradient is different than on the east side of the sound. So does that answer your question?
Oh, yeah.
Well, thanks so much, Kristen.
All right. Thank you.